Hello friends, welcome to some biology class. Friends, today we shall discuss about the principle and applications of ion exchange chromatography. Before that, I request all of my friends, please subscribe my YouTube channel and watch and like my videos and also share them with your friends. Ion exchange Ion exchange can be defined as the reversible exchange of ions in a solution with ions that are electrostatically bound to an inert support medium. The governing force in ion exchange reactions is the electrostatic force of attraction. Is the electrostatic force of attraction. Ion exchange separations. Ion exchange separations are carried out usually in columns packed with a suitable ion exchanger. The ion exchanger is an inert insoluble support medium. There are two types of ion exchangers. One is cation exchanger. One is cation exchanger and the other is anion exchanger. Anion exchanger. In case of cation exchanger, the inert support medium is covalently bound to negatively charged functional groups. Negatively charged functional groups. Whereas in case of anion exchanger, the inert support medium is bound to positively charged functional groups. Positively charged functional groups. The ions that are electrostatically bound to the support medium are called counter ions. Ion exchange chromatography is useful for the separation of charged compounds. Two main types of materials are used for the preparation of ion exchange resins. One is polystyrene and the other is cellulose. Polystyrene is prepared by the polymerization of styrene and divinyl benzene. Polystyrene resin is useful for the separation of low molecular weight compounds. Strong cationic exchangers are prepared from polystyrene, for example, by sulfonation, in which a sulfonic acid group is attached to nearly every aromatic nucleus. Weak cationic exchangers are prepared by attaching carboxylate groups to the benzene ring in place of sulfonic acid group. Strong anionic exchangers are prepared from polystyrene-based resins by treatment with tertiary amine. Secondary amine can be treated with the polystyrene for the preparation of weak anionic exchanger. Second group of material is cellulose, which is useful for the separation of high molecular weight compounds. Two main types of cellulose-based resins. One is carboxymethyl cellulose. One is carboxymethyl cellulose, and the second one is DAE cellulose. Cellulose is the two main types of cellulose-based resins that are useful in different applications. As already mentioned, ion exchange separations are usually carried out in column mode. This is a column which is packed with a suitable ion exchangers. The ion exchangers can be covalently bound to positively charged functional groups in case of anion exchanger, or to negatively charged functional groups in case of cation exchanger. In ion exchange chromatography, the sample solution is allowed to percolate through the column for a specific period of time, which is sufficient for the following equilibrium to be achieved. The overall equation for the ion exchange is E minus X plus Y plus gives rise to E minus Y plus X plus. In this equation, E minus represents the cation exchanger, and X plus is the counter ion, and X plus is the counter ion which is attached to the negatively charged exchanger by the electrostatic force of attraction. Y plus is the charged compound present in the sample. We want to isolate Y plus from a mixture of compounds. When the sample solution percolates downward through the column, the charged compound Y plus will exchange site with X plus. As a result of this, counter ions X plus will be displaced and charged compounds Y plus will bind to the negatively charged ion exchanges. So there are exchanges between Y plus and the counter ion X plus. Whereas other compounds present in the sample mixture that are either negatively charged or uncharged, they will not be able to bind to the ion exchanger, and as a result, they will easily come out of the column. Because of this exchange, the charged compound will bind with the exchanger and the counter ions will be displaced and it will be present in the solution. Like other chromatographic techniques, ion exchange chromatography also involves the use of two immiscible phases. One is the stationary phase and the other is the mobile phase. In case of ion exchange separation, ion exchange resin is the stationary phase, and the mobile phase is a solvent of suitable composition. This figure illustrates the basic process of ion exchange. These are the columns of specific size. The columns are packed with ion exchangers resin. In this figure, we have shown only a single bead of ion exchanger for easy understanding. The ion exchanger resins are covalently bound to negatively charged functional groups. Hence, these are the cation exchangers. Such type of exchanger will be useful in the separation of positively charged compounds from a sample mixture. In this example, Y plus ions that are bound electrostatically to the negatively charged functional groups are the counter ions. We want to separate a charged compound X plus from a sample mixture. The compound is present in positively charged form, so we can separate it by cation exchange chromatography. So when we apply the sample mixture on the top of the column, the sample components will migrate downward. During downward migration, the charged compound X plus will exchange site with the counter ions Y plus. So the counter ions will be displaced and the charged compound X plus that we want to separate from the mixture will bind electrostatically to the ion exchanger. Whereas other compounds present in the sample mixture that are either negatively charged or uncharged, they will not be able to bind to the ion exchanger, and as a result, they will easily come out of the column. After this, there are two procedures for eluting the charged compound out of the column. In one case, the charged compound X plus, which is bound to the exchanger by electrostatic force of attraction, can be eluted out of the column by using a buffer solution of greater pH. When we apply a buffer solution of greater pH on the top of the column, the buffer solution will migrate downward through the column, and due to its effect, positively charged compounds X plus will be converted into uncharged form. As a result, these compounds will be dissociated from the ion exchanger, and after that, they will easily come out of the column. The charged compound X plus can also be eluted out of the column by using a solution having greater concentration of counter ions X plus. When we apply a solution having higher concentration of counter ions on the top of the column, because of greater concentration, the counter ions will displace the charged compounds X plus from their site. So the counter ions will bind to the ion exchanger, and the charged compounds they will no longer bind, and after that they can be easily eluted out of the column. So in this way, different types of positively charged compounds can be separated from a mixture. For separation of negatively charged compounds, anion exchange chromatography will be useful. Ion exchange chromatography has several applications such as amino acid analysis, amino acid analysis, determination of base composition of nucleic acids. Next one is water purification, Next one is water purification and preparation of preparation of ultra pure metal ion free reagent. Apart from this, ion exchange chromatography is also useful in the separation of many vitamins, biological amines, biological amines, organic acids and bases. Friends, this is all about the principle and applications of ion exchange chromatography. For more updates, subscribe my YouTube channel Sam Biology Class.